So it's an honor to get to present to this group about the experience that we've had in trying to ramp up from a cohort study to a clinical trial for a rare inherited genetic degenerative disease. I think I'm speaking on behalf of the Dominantly Inherited Alzheimer Network that has done this. So this is not my creation, but one of a group of really talented people here at Washington University that I've been privileged to share the experience with. I hope that it will be of some use to this group who have a whole set of disorders that it looks like are prime for investigation and development of therapeutics. So I was cautioned not to spend my time teaching you about Alzheimer's disease. You don't want that. And so I'm going to avoid it, but just to frame the challenge that we had. This is a rare form of Alzheimer's disease that represents less than 1% of all Alzheimer's disease cases. But it is a critical thing because it reflects very closely the sporadic disease, which is such a major health problem in the world. The condition is actually a set of conditions. There are at least 3 genes involved that we've identified: APP, presenilin-1, and presenilin-2, with multiple mutations in those genes that result in a pathologic disease that has a very strong dominant heritability, such that in half of the offspring of affected patients, there's virtually 100% chance of developing dementia, which is severe, occurs at a younger age. So these are people that start their disease in their 30s and 40s commonly. Easier to study because they're less complicated by other aging conditions. And importantly, within a mutation, there's a very predictable age of onset, which is a huge advantage in designing clinical trials. So the effort to study this really was the brainchild of Dr. John Morris, a senior Alzheimer researcher here at Washington University, who created an NIH-funded international observational trial that was launched in 2008. And this trial, or this not trial, but it was a study, provided longitudinal evaluation of members of the families at risk. It was a global group, including sites in Canada, Australia, and in Europe, as well as the United States, and very careful, organized collection of cognitive progression, CSF and blood biomarkers, imaging, including PET scanning and MRI scanning, and nurturing the network of investigators and families was the agenda that's been followed from the inception. You can see the distribution of this network, which has continued and has expanded with support in some of other countries around the world. And as we build into clinical trials, we're in the process of expanding that network even further. Because of the rarity and widespread global nature of the mutations, it's really important for us to throw this wide net to find the people that we need to study. And I realized I'd cut out the size of the cohort, but we've accrued over 500 family members into this cohort study worldwide and followed them now ever since 2008. And the cohort has been extraordinarily valuable from a number of perspectives. Of course, it's a source for future participants in clinical trials, but most critically, it has allowed us to really exquisitely outline the progressive nature of this disease. And one of the important things that we've demonstrated that we can do is to align the progression of the disease with the expected year of onset of dementia. So we've got different genes that have different expected years of onset, but we know within a few years when in a particular family to expect dementia. So we've lined up the time that we begin studying these people at risk for or with the disease and where their family expected year of onset is. And we can lay out a really beautiful description of the disease. Here's the onset of dementia, carefully measured with quantitative testing. And you can see that we start to see the blue line is the family members, the brothers and sisters of the people with the mutations who don't have that mutation. So a very wonderful control group. And look how flat the cognitive performance is of that group, whereas the deterioration in function takes off 10 years before the onset of recognized clinical dementia when you're measuring it closely. So we were able to describe this clinical course with testing. Here, we can see the manifestations on imaging, the quantitative hippocampal volume. See how there's some aging change, but as you get 10 years or so before, you can see this deviation where the mass of the brain begins to fall away in critical areas, and the metabolism, the activity of the brain. Again, we can look at brain metabolism and see this early change that starts well over 10 years before the onset of dementia. And then, of course, there are the biomarkers marking Alzheimer's, the deposit of amyloid beta, which starts 20 years before. So we have this elegant picture of the disease that we can use to explore that and to build the biomarkers to follow in a clinical trial. So critical contribution to this design. Now, what kind of a trial did we seek to design? Well, recognizing how rare these patients are, efficiency is utterly critical. And one of the things we decided was that we would try to minimize the number of people that had to take placebo. This is a friendly thing to these patients who are facing a devastating degenerative disease, and there are no treatments at present for it. So the maximum number of people that have a shot at changing the course of the disease is what everybody wants. So a parallel design, we randomized 3 to 1 between active drugs and placebo, and we're able to get the FDA to agree to share the placebos between groups to maximize that opportunity. And furthermore, we thought, things happen in drug trials, toxicities occur, unexpected problems occur. And so if we have multiple arms going, we have a better shot to be able to carry through and really make a difference for our population. We recognized that an adaptive design was going to be critical. And so we start with a biomarker study where we start the drugs and have a picked biomarker based on the mechanism of the drugs. We have 2 drugs, they each have a different mechanism, and so we have different endpoints for the 2 active arms of the study. But we go, and if we hit the biomarker endpoint, then continue on to extend the trial for a cognitive endpoint so that we don't have to stop, re-enroll, restart the study. We want efficiency both in time and people. And as it turns out, we had to adjust the dose midstream in the study as more data evolved about what dose was going to be required. So we tried to keep going rather than lose people and time in the study. And we used all that we knew about the course of the disease to enhance the statistical power of small numbers to predict an outcome. And so we used this very sharp knowledge of the disease progression to model what we expected people to do at their point in the disease, and brought in adaptive trial consultants in statistical design, the Berry consultants, to help us to maximize the power of the study. We also realized we had to keep these people in and active, so we have very active engagement with the families, discussing what is important to them in the design of the trials, and keeping them on board and really partners in the research. One of the things they told us is they didn't want to have genetic unblinding. So that's a very important issue for their compliance. They didn't want to sign up for a trial where they had to learn what their genetic status was. And so we enrolled people that might have the mutation, but didn't insist that they be unblinded. Again, we maximized the number that could get it. Furthermore, these are young people with families that are carrying on active careers and lives, and they're scattered all over the globe. To give them their treatments, we used home nursing to go to their homes so that they would not have to take time to travel to centers performing the trial. And we really boiled it down to an annual visit at our specialty centers, which turns out to be a 4 or 5 day visit where we do 3 PET scans, multiple MRI scans, dose, and measure the extent of the disease very closely. But the rest we try to carry out at home with home nursing. So here's the design of the trial. Phase 2-3 study, we ended up recruiting 194 to get the power from just 138 mutation carriers to go to an endpoint in a 4-year trial for this disease. 2 drugs with different mechanisms. This is gantenerumab works on the deposited amyloid, and solanezumab is a soluble amyloid antibody. And it turns out that we had to run the trial 7 years for the people that first entered the trial, and the average on trial was 5.2 years in order to have a total trial duration with everybody getting 4 years of therapy. That was the design of the trial. Other important things, we found that it was critical in multiple ways to be engaged with the regulatory groups, both in Europe and in the United States. And so we had a very interactive process working with the FDA and EMA. We consulted them prior to the design. We got their buy-in to things like the shared placebo, the endpoint using disease progression modeling to look at cognitive endpoints, the introduction of dose changes and so on. They were very helpful. And one of the things that they gave us that was critical, that is important, is because of the rarity of the disease and the importance of this kind of big trial to a pivotal endpoint, they agreed that if we had a strong result, that they would consider giving a label to the drug for treatment, which was very attractive to Pharma as well as to our patients, to realize that really if we carried this through well, that there was a possibility that they could have a regular treatment that would be recognized by regulatory agencies. So important to interact with and use the regulatory agencies who are eager to help people in our position with rare diseases. Resources are critical for this, and we built public, private, and philanthropic support. You got considerable support from the NIH in the United States, which is critical so that the data and specimens and everything would be open access at the end of the day, and provides some independence and academic rigor to this. It was critical to have philanthropic support. The Alzheimer's Association and others gave significant amounts to make this trial happen. But of course, pharma support was also very critical. And we had 2 companies, and we had to learn to help them to work together, but to silo issues that were important to their intellectual property. And so we became the trial sponsor. So it's rare to have a big international trial like this sponsored by a university, but we did that. So we held the IND, but had wonderful support and were able to work with both companies to protect their intellectual property, but to make progress in the disease and trial development. So that was really critical, and it really takes a village to do this kind of massive trial. So we had a lot of people involved in the trial itself, and a bigger group of the pharma companies that have efforts to develop therapeutics in Alzheimer's disease. And so we developed a pharma consortium that these companies donated to us so that we could do the development of trial readiness activities, and gave us an additional view into the pharma world that was critical. So another part of doing this kind of trial is how do you select the drugs out of the possible drugs or interventions? How do you pick the right ones to invest this huge 7-8 year investment that we've done? And so to do that, under the Pharma consortium, we've developed a therapeutic evaluation committee. We invite companies to nominate compounds that they would like us to study and to state, sort of what their status of development is and their strengths and what the company has in mind for them. But we've put together this therapeutic evaluation committee, which is an international committee of experts, very carefully vetted for conflicts of interest, and to ask them to do an in-depth evaluation of nominated compounds, looking at their status of clinical development, likelihood of efficacy based on all the animal and preclinical data that's available, the expected side effect adverse event profile, the commitments of the companies, and to grade them on their readiness for trial. Out of the nominations that are deemed ready for trial, the Diane TU leadership begins negotiations with these companies to see if we can firm up a commitment to do the trial. So quite a complex process, very critical to pick the best opportunities and to work with the companies in a constructive way. And so I think that's a critical part of doing this kind of thing. Well, it's quite unprecedented in my experience for a university to sponsor research at this level. And Dr. Randy Bateman has just been masterful in his development of this group. And this is sort of the local university leadership that's needed for the trial. But it's just the tip of the iceberg of what we have to manage to operate this clinical trial. And so this is sort of the network. And obviously I could talk for the whole 20 minutes about this, but it's just important to realize ultimately for a global trial of this sort, you can't do it as an academician. And so we've hired IQVIA, a CRO, to assist us in the global activities that it takes to do a regulatory trial that puts a drug through rigorous testing that would make it approvable by regulatory agencies. But we have, imaging centers, quality control centers, all of the biomarkers and so on have their own groups. We have our informatics group and biostatistics aided by this global design group. And so it's really quite a village that it takes to run such a trial. But remarkably, it's happened very smoothly and done well. The other thing I just have to emphasize is how critical it is to maintain your relation with the patients and families. We found the patients through our cohort, but had to develop an expanded registry, an opportunity to screen and find other families and bring them into the group. But we maintain contact with regular webinars and patient meetings, and this is something that's been really successful. At the international Alzheimer meetings, we actually get funding to fly in the families, the patients and significant others, meet for a full day, sponsored by our trials, to really inform them of the status of the work and the issues, and to listen to them about what they're concerned about. This level of investment in the families has been critical to the success of the trial and highly appreciated. And I really think that when you're working with a tiny group of people that you're so reliant on to carry this out, you can't treat them too well. And we've really tried very well to do that. And it's resulted in a successful trial. We completed 100% of the enrollment goals, had a quite low screen fail rate as we built the study, and we've only had 7% dropout per year over a trial that's lasted a total of 7 years altogether. So a 73% completion rate with an average duration of over 5 years in the trial. It's spectacular. And what's even more spectacular is the compliance of people has been 99%. This includes lumbar punctures annually for this, and getting imaging every 3 months at local MRI sites, and coming to our centers for 4 days for, 3 PET scans in a row. It's just phenomenal, the success of running this, and it's applied to all of our arms. And so that's been a huge success. Now, if we'd had the meeting in March, I couldn't have showed you this, but this is the early results of the trial. And, I'd have to talk a long time. The sad thing is that after all of this, with the need to increase the dose in midstream and certain issues with our cognitive battery, we were not able to show a cognitive benefit of either of the interventions that we had. However, our biomarker studies really have us quite optimistic that we're on the path to something successful. And I'll just show you, these are the total tau. It turns out that CSF tau rises. It's a downstream thing. We weren't treating tau in any way with our intervention, but downstream from amyloid is the elevation of tau. And you can see in our placebo group, tau was rising through the study, but in our treatment group, it was very substantially falling. And similarly, the evidence of neural damage reflected by CSF NFL clearly was separating from our group. So we're optimistic. We're moving on to an open label trial here. And I just would share our platform. We've got an ongoing, we're developing new arms to address directly tau in the next set of therapies. And we've received an NIH grant to largely fund these and are selecting the drugs for the next arms of the study. And in parallel, are starting an even earlier intervention to block the formation of amyloid even before it starts. So it's really possible to do this with talented leadership. That's not me, that's Dr. Bateman, Dr. McDade, and a really terrific team that's built around a strong cohort study. And so this group of centers is spectacular. And it's a really exciting academic and clinical enterprise that I recommend you try for your diseases as well. And with that, I will stop.
The IDEAL project, my work currently is involved in the European Joint Program on Rare Diseases, where we demonstrate the use of the methodologies we developed in the IDEAL project, as well as we would like to develop new methodologies which are tailored for clinical trials on the one hand. So my talk is more conceptual from the biostatistical part of view. I don't have so many examples right now, but well, my basis is a little bit around that. So from Rima Nabout, my colleague in Paris, whom I'm leading the work package 20 in the EGPRD. I had discussion about Dravet syndrome, where a parallel group design clinical trials has a primary endpoint of 50% reduction in monthly seizures. And I found this when I read the paper that it is very insensitive, because it's an averaging process, the 50% reduction. And I discussed with her about counting the seizures over the time, rather than doing something like 50% reductions. So that brought me to another application area where I was involved in, is the way of the Friedrich ataxia, where Jörg Schulz and Katrin Reitz involved me in the analysis of the registry data. We made an evaluation for the 2-year trials, and now for the 4-year trials it is in progress. And the progression of SARA score was a little bit what I found more interesting than doing baseline to the difference in baseline to a 2-year value, because if you model the progression, it can be more sensitive. And we do that for SARA scores, CAFI, ENAS, and as well as ADL. And with these data in mind, we planned the clinical trials where placebo is compared to nicotinamide, and this trial will start soon, and we made the planning of the trial with a progression rate in SARA score and something like that. So basically, the last two examples were brought to my mind by Mattis, and these are well, this brings me to questions. Well, do we have methodologies which are tailored for really limited populations, where you have a limited number of patients which you can involve in your trial, and do we have something for this? So basically, to answer these questions, you see in my title that I'm talking about ultra-rare disease, which I cannot define really very specific. I think we should go from three directions on. First, we should look to alternative primary endpoints. Second, we should include, let's say, past data or information we have and use these in clinical trials. And the third one is, when you are really in a limited setting, you need other approaches. What I would like to start with is with rigor use of longitudinal model methodologies to describe this progression, and I give you some examples and some problems which are also can be solved but are less known right now. And then I will go to sample size calculations. So what can we do in that direction? What should we be aware of? And then I will go to the second part with including historical controls and the evidence of level of evidence. So already in the effects data, we established the linear mixed effects models. And if you have slopes, progressions like that one, which you see here for the SARA score, it's a black line, and for the subscores in the colored lines, which are almost linearly, then linear mixed effects models provide a sufficient solution to model the longitudinal data. In particular, in the cases when you don't have any flawed or sealed observations. You might ask what is depicted on the y-axis. Well, we normalized all the scores by dividing by the highest score, and that is why they could be drawn in one in one figure. But well, let's make a thought experiment, and let's think about the sitting subscale of the SARA score. And you are fine if your patients provide data like that, which I depict here. You have at a particular time points, let's say 4, 5, 6, 7, and 8. You have scores of 0, 1, 2, 3, 4, and then you will do a linear regression to that, and all is fine. You can use standard methodologies to establish the progression. But unfortunately, life isn't like that. What you see is in these sitting scores that you have sealed observations in your data. So some patients, if you prolong the observational period, stay at the 4, and they will not go down, they will stay there. And well, you have the impression that the red line is not a good representative for the slope of these data. And you might ask, well, what can I do? Should I skip the flawed or the sealed observations? And what you also see is, as more sealed observation you have, as more your progression line is deteriorated. And that is not a nice picture of what you have in your data, and makes probably your response variable to a treatment affected negatively. And the same holds true if you also add flawed observations to that. You cannot use standard regression theory. So that's what it is at the end. So what can we do? Well, we can use other statistical methodology which are known in some areas as centered regression model, and it is represented by the blue line. And this centered regression model uses the sealed and the flawed observations, but tried to do to account more the data which are in the progressive part of your data. And you see it here below. If you use a linear mixed effects model, you have a slope of 0.35, and with the centered regression model, you got 0.29. And these so-called centered regression models, also known as Tobit models, because Tobin has developed this methodology in 1958, but nowadays we are possible to make the calculation for these models. These can be used for modeling these, let's say, subscores of the SARA score. And you might ask, well, is there a difference? Well, in your figure, I see the difference. But when is there a difference between the standard linear effects model and the Tobit model? Well, you need to have sealed observations or flawed observations at the particular time points higher than 15%. That's more my experience than it is a validated statement. What we have right now, because there's no investigation about that. So let's go one step ahead and let's do sample size calculation with this with the linear mixed effects model, which cannot be done right now with standard statistical software. You need to simulate that. Well, I have written some software package for this based on SAS routines. And I did that also with the EFAX data. And interestingly, you can find two things. Well, compare the red line, the blue line, and the green line. They are different in trial duration. So red line is a 1-year trial, blue line is a 2-year trial, and green line is a 3-year trial. And what I changed on the x-axis is the number of the frequency of visits of my patients. So this means, the 4 means that you have 4 months, that you have every 4 months a visit of your patients. So this point here in the 3-year trials with 12 months visit corresponds to the 4 months visit in a 1-year trial. And you see that you can earn in your trial if you prolong your trial, you make it longer with the linear mixed effects model, and you can also earn if you do frequent re-observations. But that is a very critical point. As Jörg Schulz told me one, he said, you cannot evaluate the SARA score, let's say, on a monthly basis, because there is so many learning effects in this score that you will have probably wrong results at the end. So there must be found a balance in that way. So I come to my next point, using other information, using historical data. I have heard that a lot of time when I've talked with people who are dealing with rare diseases. They have registry data like that one here, NH, and they would like to use this in the randomized clinical trials. Can I do this? And will it benefit? Well, one problem occurs immediately, and the problem is that you have to make sure that the historical control data are similar to what you have in your randomized trials. So you have to establish that the historical control data and the randomized control data are the same. And we are evaluating an approach where we use an equivalence test for showing that the historical control data and the randomized control data are similar. And if this is true, then you can use in your evaluation of the randomized data the historical control data, and probably you come to an end. And our procedure goes one step further. If you fail to show the equivalence, then you should, in an adaptive way, recruit further patients in the experimental group as well as in the control group, and to fill both groups up, to come to the pre-planned end, and to establish the new the desired treatment effect. Well, this is under publication right now, so we hope that we can submit the publication in a few months, so that we can show you much more details. And this procedure is a balancing between the effect you would like to establish between the number of historical controls you would like to have, and all these things. And you can extend this also if you would like to extrapolate. Say, think about that you have a clinical trial in adult patients or in a similar disease area, where you always also have shown that your experimental treatment works. And now you would like to do a similar study in an ultra-rare disease, where you cannot include so many patients. So then probably you can combine all these two trials in such a way that you have to establish equivalence between the experimental data here, between the control data here, and between the treatment effect you see in the larger population. That should be equivalent to that what in the ultra-rare disease. Well, this is not evaluated right now. This is our next step we would like to do, and then we can go and tell you how to proceed in that direction. Other clinical trial setting which you can think about when you are doing clinical trials in ultra-rare diseases are so-called N-of-1 trial. So you have end treatment period in one patient. And if you plan this in advance, you can apply this in several patients, and then you can combine the results so that your statement at the end would not be limited to the one patient. It could be in some sort of way generalized to a larger group. And we have other designs. I have recently heard about a blinded start design, which is known in our statistical context as a stepped wedge design. And I will give you some warning, because there might be some residual effect in this trial which are not discussed right now as it should be. So anyway, there is some study needed to do it like that. I would like to come to the end. What can we do if we have only 20 patients in the world and would like to do something for them? You know, well, we statistically have three areas, and you can say, or categories, and you can say, I'm Bayesian, I'm frequentist, so I'm Carl Pearson statistician, or I'm Fisherian. Well, the Fisherian world has lost for a long time, but I think that is a very good opportunity to do an experiment where you can make a comparison if you have, for example, only 8 patients which you can involve in a clinical trial. And I discuss this right now with a biostatistician at Novartis, Alex Svetlov, and I have on Friday a meeting where we would like to plan a clinical trial like that. So it is done partially, as Jopi reported in a report, where a clinical trial with n equal 8 in total leads to the legislation of a drug. So that's incredible. But the EMA has legislated that. And well, if they are aware of similar thing, then we have to ask, well, this population model like Bayesian or the usual Fisher, the usual t-test, as you know, we put a lot of assumption in these approaches, and you do not need this in the Fisherian world. What you do is you select an appropriate randomization procedure, and if you select that, then you can learn from your data a lot. But what is an appropriate randomization procedure? Most of the people think that permuted block randomization is the best you can do. And I show you that it is the worst you can do, because this procedure, the cumulative distribution function, is like that. And you see others like random allocation rule, which is the green one here. And the big stick design, which is the blue one here, has some profit. So my last comment, yeah, I have to stop, to come to the end. My last comment is that you can profit by using other randomization procedures, and you can also establish the 5% level. And that's what I would like to say in that. So my comment is that randomization-based inference could be used in ultra-rare diseases, should be soundfully planned, and then probably you should go to the regulators and say, okay, that level of evidence we can get from my data. So with this, I come to the end. I'm sorry about the confusion with the presentation at the end. Sorry about that.
Thank you, Ralf, for your wonderful presentation. We learned a lot. I invite everybody to go to the coffee break to present questions to Ralf as well. And now we will hear Dr. Jane Larkindale. She's, She's executive director of two initiatives that belongs to the Critical Path Institute. We all learned yesterday from home talk that Critical Path Institute will help SCA Global and ARCA in understanding and preparing outcome parameters for clinical trials. So now we will hear what she can tell us about it. Please, Jane, take your time now, 15 minutes if possible.
Wonderful. Thank you very much to the organizers for giving me the opportunity to speak today, and I'm very happy to be here with you. I'm going to cut some of my earlier stuff just because I think it's been covered already, but for those of you who don't know the Critical Path Institute, we've been around about 15 years. We were formed by a memorandum of understanding with FDA, really with the goal of delivering on what the FDA called the Critical Path Initiative, which was an initiative to try and accelerate drug development broadly across all diseases. So what we do as an institute is we focus very strongly on developing tools to accelerate drug development. We have a number of programs. We currently have 16 different programs. We're a relatively small organization internally, but we collaborate with over 1,600 scientists and 90 organizations worldwide, because everything we do through collaboration and community consensus. We don't do everything ourselves because let's face it, we're not experts in every disease or every area that we work on. The areas of expertise that we really have are the regulatory science and the development of drug development tools. So all of our different programs are focused on developing things like biomarkers and outcome assessments, mathematical models like Ralf-Dieter was just talking about, innovative analyses to really move drug development forwards. And you can see a number of our programs are specific to individual diseases. So we work on a number of neurological diseases. We also have a number of programs that are focused on specific types of tools, such as safety biomarkers or patient-reported outcomes. The two programs I have highlighted here are our Friedreich's ataxia integrated database, which is obviously of interest just because this is an ataxia, and our rare disease cures accelerator, which is a data and analytics platform across all rare diseases that I'll talk about a little bit more later in the talk. These are probably the two current programs most associated with the ataxias. But as Holm has already mentioned, we are in the process of launching a pan-ataxia consortium to specifically focus on the ataxias. So that's new. It's not on the chart yet because we don't formally exist yet, but it's well into the planning stages, and we hope to formally launch soon. So this slide really just summarizes the work that we've done over the past 15 years. Pretty much all of the programs we do have a goal of reaching some sort of formal regulatory acceptance of a tool, whether it's a qualification decision, a fit for purpose endorsement, or some kind of letter of support to really suggest that regulators will accept the tools for a specific context of use. I will not go through the slide in detail, but these are some of our qualified biomarkers, outcome assessments, and models. So there are several different ways by which you can reach acceptance of drug development tools. These slides really come from an FDA presentation, but it's true also for EMA and other global regulators, that we can work through an individual drug development pathway. That's obviously one standard way of doing this, so that you can look at one particular tool for one particular drug within the context usually of one company, and that's fine, that's one way of doing it. But the way we typically work are more globally across disease communities. As I say, we tend to work through a consensus-based approach for an entire disease community. So we tend to work through the qualification programs. These are formal programs that they have at both FDA and EMA to really review and accept data around a particular tool and qualify it generally for a specific context of use. And I'll talk quite a bit about what those programs are like. And then the third method, which may in many ways be the most appropriate for rare and ultra-rare diseases, is what they call scientifically supported community implementation, which is when an entire community has been using a tool for so long and there's so much data behind it that although it may not have a formal endorsement, everyone's agreed it can probably be used for a specific context of use. And I'll talk about that also and what level of information we would need to get to that point as well. But let's take a step back, and I think Massimo covered this to some extent yesterday already. What should an endpoint assess? By the FDA definition, an endpoint assesses how a patient feels, functions, or survives. So that's pretty obvious. That's why we take medications. We want to know whether how a drug would affect our survival, or how it will improve function, or how we feel. There are many different types of COAs, but in contrast, a biomarker is a characteristic that's an indicator of either a normal biological process, a pathogenesis process, or a response to exposure or intervention. So I'd like to include those definitions just so we can be clear about what we're talking about. There are different types of clinical outcome assessments. That's the way a drug would typically be approved. And there are different types and context of use for biomarkers, which is not typically a way that a drug will be approved through standard pathways. But if enough data exists around a biomarker, it can be used for an accelerated approval. And I think across the ataxias, there's a great deal of interest in both improving outcome assessments and biomarkers. So let's take a step sideways and look at what these qualification programs are like. What is FDA or EMA looking at to allow us to qualify and really accept drug development tools? The FDA pathways are called the drug qualification pathways. There are 3 different qualification pathways, one for biomarkers, one for clinical outcome assessments, and one for animal models that I won't be talking about much here. Essentially, qualification is a conclusion from the agency that within a stated context of use, the drug development tool can be relied upon to have a specific interpretation and a specific application in drug development and review. So you don't have a biomarker or an outcome assessment qualified globally. It's qualified for a specific context of use. And this is really important because depending on how you want to use the biomarker, you're going to need different levels of evidence. If you want to have a biomarker used for as a surrogate endpoint for approval, you're going to need a whole lot more evidence than if you want to use the same biomarker to define a population to enter a trial as a prognostic biomarker to determine who's going to have a particular outcome over time. But the same biomarker with more evidence may be able to be used as a surrogate endpoint. So we need to think about these things. The EMA process is similar. They have a process called the Qualification of Novel Methodologies in Medicine Development. The difference is that this is one pathway where you look at COAs, you look at biomarkers, and you can look at mathematical models of disease and other tools as well. It's a somewhat more formalized process in that it's got very distinct timelines of submission, but we at CPath tend to use both pathways as much in parallel as possible. So in terms of how to go about developing COAs, I'm just going to refer you to this guidance rather than reading through the slide in the interest of time, but there are very distinct questions one needs to answer in order to develop a regulatory grade COA. And you need to understand the disease, what's important to patients, the natural history, what's going on in the patient population. Then you have to identify what they call a concept of interest, of what it is that you want to measure. What is aspect of how a patient feels, functions, or survives you're trying to measure? And then from there, you can define your context of use, and then move into actually thinking about what the COA is. Is there one already in existence? Do we need to develop one to move forwards? And if you're getting a regulatory review of a clinical outcome assessment, they're going to review the data at all of these stages. Biomarker development is similar, but not exactly the same. Instead of a concept of interest and quite so much data on why this is relevant to patients, you spend a lot more time thinking about the analytical validation of the biomarker and the clinical validation of the biomarker. So understanding the performance characteristic of the test and understanding how that biomarker correlates with outcomes of interest, and really trying to understand how directly you can relate that biomarker to what it is you're trying to measure in a population. And I will tell you that some of the safety biomarker qualification packages that I've been involved in have run to 400 or 500 pages. This is a lot of data, which I'm summarizing in 4 words: analytical validation and clinical validation. There's a lot of work that's involved in this. Assuming you're going to go forwards, this is actually the timeline for FDA. EMA's timeline is similar. The process for qualification is as follows. You submit a letter of intent. They take 3 or 4 months to review it. Hopefully they say, yep, this seems like a reasonable thing to do. Then you submit a qualification plan, which describes all the work you're going to do and everything you want to put into your final submission. They take a while to review that. EMA is a little bit tighter on their timelines for reviewing that. They hopefully come back and say, yep, your plans are good. Realistically, they come back and say, your plans are good, but we also want to see these 7 other things as well. And then you go forwards and create your qualification package, which they again review for quite some time. What you'll notice here is that just the FDA review time of this is going to take a year to a year and a half, let alone the time it takes for you to also do the works. So these are not processes to undertake trivially. It's going to take a lot of time and a lot of effort, but that's okay. So long as we can get there and we can get enough data that we can get qualification, we can get the agencies to say, we can use this data, this tool for this context of use. It's a really powerful thing for the community to be able to go into a trial knowing exactly how we can use a tool without arguing it, discussing it with the agencies to move forwards. But it is a long process and it requires a lot of data. And a challenge for rare and ultra rare diseases is that it can be very, very slow, and we may not be able to achieve that level of data. So instead, the alternative is to look at this third pathway, scientifically supported community implementation. And this is really where, as a community, we come together, we really think about it, we pull all the data we have together, we probably have meetings, we may have meetings with the regulators and say, well, look, we're not going to get all the information we need, but what do you think? Do you think this is worth going forwards? So how do we get there? Really, we look at exactly the same things as we would for qualification. We just may not have quite as much data. You're still looking at analytical and clinical validity of a biomarker. You're still looking at concept validity, clinical meaningfulness, and clinical validity of an outcome assessment. And we really need to understand the change in the measure and the variance in the population of interest. And the way we've been looking at this and often, and we're looking at this in Friedreich's ataxia, and I think is how we'll look at it across the other ataxias, is by building models of endpoints and biomarkers, trying to get regulators to comment on the models, the understanding of the change, because that tells us how we can use a biomarker or an endpoint, so we can really understand that. And I was really interested in the previous talk where Ralf-Dieter was talking about some analysis that he had done. I know there's been a bunch of other groups looking at this. And I think this is a very powerful way forwards. I've got a few examples here. I see my animations aren't quite working, but that's okay. So these are examples from other disease areas where we've done this quite successfully. The first one was looking at a biomarker, total kidney volume in polycystic kidney disease. This was qualified using a modeling approach as a prognostic biomarker, really looking at the size of the kidney to predict which patients were going to go into end-stage renal disease. But over time, it was used, more data was acquired, and it's now been accepted as a reasonably likely surrogate endpoint. And a drug has actually been approved based on this biomarker. And there's a lot more analysis going forwards. This is a great example. The data wasn't great. There were actually no drug trials being done in polycystic kidney disease when this work was started. but over time, the data was acquired from clinicians, it was integrated, it was modeled, and we could really use it to move drug development forwards. My second example is not in a rare disease. This is in Parkinson's disease. Obviously, we have a lot more patients, but it was looking at dopamine transporter positive patients as an enrichment biomarker, where we built models to demonstrate how we could change the trial size by enriching a patient population with patients who were likely to really progress to clinical Parkinson's disease. The graph just shows that by using this biomarker to enrich the trial, we could reduce the number of patients needed in the trial by 25%. I think this approach is probably less useful in the ataxias because we have so few patients already. Would be more likely to try and use a modeling approach to increase the number of patients involved. And then my third example is looking at an endpoint. This is the North Star ambulance assessment in Duchenne, which was hugely variable, where we've used a number of models to try and really understand how different groups of patients will progress, with the idea of understanding which patients we should look at which stage in disease to see what level of change. You can see with the solid lines, the data, and with the dotted lines, our model predictions, which really show that our models are predicting quite well the different progression of different groups of patients and who they are. And this is early work. We're just completing our EMA package now, so it has not been endorsed yet by the regulators, but we're working on that, and it should be soon, we hope. So with that, I'm running out of time. So briefly, I just wanted to talk about the Rare Disease Cures Accelerated Data and Analytics Platform. This is a program that's been going for about a year to try and integrate data across all rare diseases. And this is where it's going to be very appropriate for the ataxias, because the idea is to bring data not just for one disease, but as many diseases as possible, curate it, bring it together, and be able to integrate it and build these kinds of tools. So if you're interested in a disease progression model for SCA3, you can look at SCA3 data. If you're interested in neurofilament light data, you can look at neurofilament light across multiple diseases. If you're interested in the dynamics of SARA score, you can look at that across various diseases. And this is the platform we'll use for data integration for the new ataxia consortium. And so I'm sure we'll have an opportunity to talk more about that soon. Just as a thank you to all the people I've worked with who've contributed data, shared data with these various programs, and of course the FDA, who is funding our Rare Disease Data Accelerator. And a great big thank you to the organizers.